Well, hello, everybody. Thank you so much for joining us here for this session on emerging signaling pathways. We're very excited to be here. My name is Marina Holz, and I'm joined here by Idil, Ulengin-Talkish, and Ravinder Abrol. They're coming from California, so a lot of thanks for logging in so early and giving their talks here. So I think without further ado, I will start my talk, and I'll start sharing my screen. All right. So I'm very excited to be here. As I said, my name is Marina Holz, and I will talk to you today about crosstalk between mTOR and estrogen signaling. I'm professor of cell biology and anatomy at New York Medical College, and I am also the dean of the Graduate School of Basic Medical Sciences. And you can also contact me on Twitter, and I think my email address has been shared with this group in the chat. My lab works on the crosstalk between mTOR and estrogen signaling. And as you can see, this is a very complex interactive network. So mTOR complex 1 is the rapamycin sensitive complex. And here I show the composition of this complex. There are several proteins that interact together to mediate the functions of mTOR C1. And this complex is acutely sensitive to the naturally derived inhibitor rapamycin. mTOR is activated by a variety of upstream signals that control various anabolic processes in the cell. In addition, what we are also interested is how estrogen, which is a lipid hormone that can cross the membrane and bind estrogen receptor to activate transcription of estrogen responsive genes. We also are interested in two questions. One is, how can estrogen activate these pathways that are thought to be usually growth factor and nutrient sensitive? And the second question is, in response to estrogen activation, how does the mTOR signaling network regulate the function of estrogen receptors? So one of the questions that we're trying to answer is, what are the downstream targets of mTOR signaling that are activated by estrogen in the cytoplasm? And the second question is, what are the estrogenic targets of mTOR in the nucleus? So how does mTOR regulate estrogenic transcription? And this figure comes from a review by a graduate student in the lab, Christina Faltas. So estrogen has very diverse effects on pathoactivity in the cytoplasm. So here what we did is we investigated various signaling pathways using a combination of inhibitors that inhibit various important signaling nodes in the cell. For example, we used the mTOR inhibitor rapamycin, mTOR catalytic inhibitor PP242, which inhibits both mTOR C1 and mTOR C2, MEK inhibitor UO126, and AKT, pan-AKT inhibitor MK2206. And we looked at two estrogen-sensitive cell lines, MCF7 and T47D, and we asked exactly what pathways are activated in these cells. And what we noticed is that there was very strong activation of mTOR signaling in response to estrogen, as well as PI3 kinase signaling. But the ERK signaling was largely insensitive to that. So there's clearly some differences. In addition, there are also cell type dependent differences if you compare the two cell lines. So there's clearly some questions here that are unanswered. In order to look into what are the global phosphorylation targets of mTOR when activated by estrogen, we used phosphoproteomics. And what we did is we had MCF7 cells, which are ER positive estrogen sensitive cells, and we treated them with estrogen, or we treated them with estrogen in the presence of rapamycin to inhibit mTOR, to specifically look at these mTOR-dependent phosphopeptides. And we harvested the cells and processed them for TMT10 analysis. And the result of the of this TMT10 screen revealed to us that there was a very small number of estrogen responsive sites that are upregulated relative to control, and these are a little hard to see with the blue dots here, and a much larger number of rapamycin sensitive screens sites that varied in different directions, and these are the red dots as well. And yellow dots are the known mTOR targets. So clearly we hit on some of the known targets as well, and many others that were unknown. And we summarize some of them in this table. And what we see is that indeed we see some known mTOR substrates and phosphorylation sites. And also we detected several known mTOR substrates with previously uncharacterized phosphorylation sites, which was interesting to us. And here I highlighted some of the things we decided to follow up on. So in red, we identify many translation initiation factors because mTOR is a major regulator of translation initiation. And also we identify mTOR here in yellow, which is an inhibitory subunit of mTOR C1. So we decided to follow up on these two aspects of the screen on translation regulation as well as on mTOR. In addition, we also looked at regulation of gene expression under similar conditions. And what we found out is a lot of the previously characterized genes, for example, GRAB1, which is a known estrogen target, as well as RAD51C, which we previously characterized in a different paper. But we also discovered DEPDC6, which is the gene that encodes for mTOR. And we also uncovered EIF3F, which I'm going to talk about in the second part of the talk, which is a translational regulator, and it was actually downregulated in response to estrogenic activated mTOR signaling. So that was a very interesting question to us. So in order to first characterize mTOR, this is a very interesting subunit of mTOR C1 because it is inhibitory. However, it is also inhibited by mTOR. So mTOR phosphorylates mTOR, which targets it for degradation. So first what we did is we wanted to confirm that in response to mTOR activated estrogen signaling, mTOR is indeed phosphorylated, and we saw that it is phosphorylated and that phosphorylation is sensitive to everolimus, which is a rapamycin analog that inhibits mTOR. What we did next is that we examined phosphorylation of other proteins in the PI3 kinase mTOR network to see how they respond to estrogen stimulation in the presence or absence of serum. So here we treated the cells with 5% FBS. And what we noticed is something peculiar, which is that the levels of mTOR were actually stabilized in response to estrogen treatment, and it was insensitive to rapamycin or mTOR inhibition, which indicated to us that there is estrogenic regulation of mTOR expression that is independent of mTOR phosphorylation. So we decided to follow up on that in more detail. We took the same conditions and we treated cells with either low serum or high serum conditions in the presence or absence of estrogen. And under all these conditions, no matter how we tried, we still were able to detect that when cells were treated with serum, mTOR was degraded because mTOR was activated and led to degradation of mTOR. However, in the presence of estrogen right here, we saw that there is stabilization of mTOR levels. So this confirmed to us again that there is this activity that upregulates mTOR expression or prevents its degradation. So what we did next is we asked whether this is dependent on actual transcriptional activity of estrogen and estrogen receptor alpha. So what we did first, we looked at the function of inhibitors of estrogen receptor, either fulvestrant or tamoxifen. So fulvestrant is a degrader of estrogen receptor alpha. You can see it right here very well. And tamoxifen is a selective estrogen receptor modulator. It prevents binding of estrogen. And what we saw here is that indeed there was a role of estrogen receptor in this activity. And in addition, we also looked at chromatin immune precipitation, and we could detect the binding of estrogen receptor to the promoter of mTOR. We also looked at it in a different level. So we know that mTOR binds and inhibits mTOR, and we try to see whether this is mediated by estrogen. And indeed, what we saw is that there is a regulation of mTOR binding to mTOR when cells are treated with estrogen. So not only mTOR is stabilized, but it is now also induced to interact with mTOR. As we detected by immunoprecipitation assay. And when we knocked down mTOR using a siRNA, we also saw upregulation of mTOR signaling. So to conclude this part of the talk, what we saw is that we try to make a connection between estrogenic and mTOR signaling, which is very important for many diseases, especially estrogen receptor positive breast cancer. And to characterize this connection, we did the phosphoproteome analysis of ER-positive MCF7 cells, and we identified mTOR as estrogen-regulated mTOR target, which is an inhibitory protein of mTOR-C1 and also mTOR-C2, and usually is degraded by growth factor-activated mTOR signaling. And what we saw is that estrogen through ER transcriptionally activates mTOR expression. So despite decreased stability, mTOR levels are upregulated by estrogen to modulate the function of mTOR-C1 and mTOR-C2. So I want to go back to this screen that we did. And as I said, we also identify many translational regulators, as well as this translational inhibitor, EIF3F. And we decided to follow up on that as well in a little bit more detail. So mTOR is a known regulator of translation, and it participates in a complex to act through two of the main regulators of translation. One of them, the EIF4E inhibitory protein, EIF4E-BP1, which is phosphorylated by mTOR and is released from this cap binding protein EIF4E, allowing the cap complex to assemble and to initiate the translation. And then the target of mTOR is this kinase S6K1 that phosphorylates many regulators of translation, including the 40S ribosomal protein S6 and translational factors EIF4B and PDCD4. And EIF3 is a scaffolding complex that mediates many of these interactions, and we're previously very interested in that. So EIF3F is a subunit of EIF3, which has about 13 known subunits. So it's a very big complex. I apologize for that. Don't we hate Adobe? EIF3F specifically shows this decreased expression in many types of cancer, specifically breast. So we decided to follow up on that because, as I said, we are interested in estrogen responsive cancers, especially breast. So first, we decided to look at the levels of different EIF3 subunits in different ER-positive and ER-negative tumors. And what we saw that in ER-positive tumors, there are actually lower levels of EIF3F compared to ER-negative tumors. So this was interesting to us because it confirmed with this notion that EIF3F is underexpressed in ER-positive breast cancer. And we decided to investigate that in a little bit more detail. So what we did is we looked at the expression of EIF3F on the mRNA level. And what we saw is that in contrast to the known estrogen responsive gene TFF1, we saw that, which is usually upregulated by estrogen, we saw that EIF3F was actually downregulated by treatment with estrogen. And we also confirmed that in terms of the stability of mRNA levels. And we also confirmed that in the presence of cyclohexamide, this was not observed. So this was not something that was dependent on protein synthesis. Then we also looked whether this is actually dependent on the presence of estrogen receptor itself. And again, we compared EIF3F expression to that of TFF1, which is a known regulated gene of estrogen. And we saw that unlike TFF1, which is upregulated by estrogen treatment, EIF3F was actually downregulated. And we saw that also when we inhibited ER expression using a siRNA against ER alpha, whereby we saw that the upregulation of TFF1 was dampened, and this negative regulation of EIF3F in the presence of ER inhibition actually went up. So this indicated to us that estrogen acting through estrogen receptor regulates EIF3F expression. We then looked at whether the binding of ER actually occurs at the promoter of EIF3F. And we had different sets of primers in the proximal promoter of EIF3F, as well as at a putative ERE, which is a bit upstream. And what we saw is that we did detect ER binding at the proximal promoter, which indicated to us that there is regulation of EIF3F expression by ER binding, and it's likely through a mechanism that is known as physiologic squelching, whereby ER binds to the proximal promoter and it removes other positive regulators of transcription from the promoter. So to come back to the role of mTOR, we know that mTOR regulates translation, and EIF3F is a translational inhibitor. And we asked whether or not estrogen, first of all, promotes cap-dependent translation via mTOR. And to answer this question, we used this dual luciferase reporter, whereby Brinilla expression is controlled by the 5' cap, whereas Firefly expression is controlled by hepatitis C virus iris. So it's independent of the cap, and by comparing the two, because it's a bicistronic promoter, we can actually compare the effects on cap-dependent translation. And what we detected here is that in the presence of estrogen, there was indeed increase in this translation of this promoter, and it was rapamycin sensitive. And not only that, it was also sensitive to anti-estrogens such as fulvestrant and tamoxifen. We then asked whether EIF3F overexpression can suppress these effects, and indeed it could. So using the same dual reporter system, we're able to observe increase in the activity of the cap-dependent promoter with estrogen, which was dampened when we overexpress EIF3F, which is the putative inhibitor here. And then we also asked what are the physiological effects of EIF3F overexpression. So when we overexpress EIF3F, we also observed that there is increase in apoptosis of the cells overexpressing EIF3F and decrease in proliferation. This was actually a tricky experiment to do because when we overexpressed too much EIF3F, all the cells died. So it was very hard to do any kind of time course. So we really had to do sort of a transient transfection with transient G418 selection in order to find that window where the cells just died more slowly than under other conditions. So to summarize, what we observe here is that when we have unstimulated cells, EIF3F transcription is up and it downregulates protein synthesis. But when estrogen stimulates cells, there is a need to increase protein translation in the cells and increase protein synthesis. And in that case, EIF3F is suppressed in order to allow for protein synthesis to happen. So this is the end of my talk, and I would like to thank the talented people who work in my lab. So most of this work was done by a former postdoc in the lab, Rafa Cuesta, who now moved on and is at McGill University in Montreal. And this work, I should mention, was also published in 2 journals of ASBMB, JCB, and MCP. So I highly recommend, this is a plug. And also other people who are here in the lab with me. We're slowly ramping up our research operations. So this is really great. And I want to thank my collaborators at Pacific Northwest National Laboratory who helped us with the phosphoproteomics, as well all of my funding sources, past and present, and of course you.
Okay, we've gone a little over on time, so Ravinder, could you just ask one question and Marina can answer the rest by typing them in?
Okay, sounds good.
Thank you.
Thanks, Marina. It's a great talk. I'm always impressed by the complexity of signaling with many crosstalks. So the first question that came was, is DEPTOR phosphorylation mandatory for estrogen-based regulation of DEPTOR-mTOR coupling?
So DEPTOR is, this is a great question. So DEPTOR is regulated by both growth factor signaling, as we previously known, and now we know by estrogen signaling. So there appears to be a modulation depending on which activator or stimulant is present in the cells at that particular moment, or the relative quantities of these activators. So in the presence of growth factors, DEPTOR will be primarily degraded, and in the presence of estrogen, it will be stabilized. Thank you so much, and I'll answer the rest of the questions in the Q&A chat.
Yeah, thanks, Marina.
So we are next have our next talk, and I'll stop sharing my screen so the next presenter can do that. So our next presenter is Idil Ulengin-Talkish from Stanford University. And she will talk to us about discovering unique functions and regulation of palmitoylated calcineurin isoform CNA beta 1. That's amazing, so take over.
Thank you, Marina, and I would like to thank the organizers for giving me this opportunity today. I'm very excited to share with you the novel roles and regulations of the protein phosphatase calcineurin that we have discovered through studying an isoform of calcineurin that has been virtually unstudied, despite the fact that it is expressed in every human tissue and it is highly conserved among vertebrates. Calcineurin is a serine threonine phosphatase and a key regulator of calcium signaling with multiple critical functions important for human health. Calcineurin is activated in response to the increased intracellular calcium and calmodulin levels, for example, as a result of calcium signaling from ligand-induced GPCRs. Activated calcineurin then dephosphorylates specific substrates, leading to regulation of many cellular processes and thus downstream propagation of the calcium signal. Calcineurin is most known for its role in dephosphorylating the NFAT family of transcription factors, which then translocates to the nucleus, where it activates the expression of genes important for T-cell activation and proliferation. Indeed, it's this role of calcineurin in activating the adaptive immune response that makes calcineurin a critical target, where immunosuppressant drugs that are prescribed to the organ transplant patients work by inhibiting calcineurin's activity to prevent organ rejection. But today I'm going to introduce a much less well-studied calcineurin family member, which does not activate the NFAT transcription factors. And so far, its functions and substrates have not been discovered. Calcineurin is a heterodimer that's composed of a regulatory B subunit and a catalytic A subunit that is encoded by 3 genes, and the alternative 3' pre-mRNA processing of the catalytic beta gene gives rise to 2 isoforms, the canonical CNA beta 2 and CNA beta 1 with a non-canonical C-terminal tail. Each isoform share the same catalytic domain, but the C-terminal tail of CNA beta 1 is dramatically different due to translation of intronic sequences. Here is the tissue distribution of human calcineurin isoforms derived from RNA-seq data. You can see that CNA beta 1, which is shown in green, is expressed in every human tissue alongside with the canonical isoforms, albeit at much lower levels. But although it is expressed in every tissue, it is very understudied and not well characterized. The few published studies of CNA beta 1, however, demonstrate that it has distinct physiological properties. For example, overexpression of the canonical isoform CNA beta 1 in mouse cardiomyocytes is found to activate NFAT, which causes hypertrophy. In contrast, overexpression of CNA beta 1 did not activate NFAT and was found to be cardioprotective. And indeed, recently it has been shown that the CNA beta 1 knockout mouse actually develops cardiac hypertrophy. And furthermore, in vitro studies done by our lab revealed that CNA beta 1 enzymatic regulation was also different. It was found that it was only partially activated with calcium and calmodulin due to a distinct mode of autoinhibition conferred by its distinct C-terminal tail. So these studies suggested that this isozyme has a distinct properties, however, did not provide a substrate or mechanism of function of how the CNA beta 1 confers these properties. And this left us with more questions to answer. For example, does CNA beta 1 have specific substrates? And if so, what is the mechanism that confers this substrate specificity? Next question is, how is CNA beta 1 regulated in vivo? Are there additional mechanisms that regulate the enzymatic activity of this isozyme? So the biggest clue came when we expressed the isoforms in mammalian cells. Overexpression in HeLa cells revealed that unlike the canonical calcineurin isoform CNA beta 2, which is cytosolic, CNA beta 1 isoform was membrane associated, and localized to the plasma membrane, as well as Golgi and intracellular vesicles. To understand how CNA beta-1 is associated with the membranes, we turned to its unique C-terminal tail and determined that these two highly conserved cysteines get palmitoylated using multiple approaches. And here I'm gonna share with you one of them, the acyl rack, that allows the capture of palmitoylated proteins by binding of their modified cysteines to the beads when exposed by hydroxylamine that cleaves off the palmitate groups. We saw that CNA beta-1 is palmitoylated similar to the our positive control palmitoylated plasma membrane protein EFR3B. And the canonical isoform of calcineurin is not palmitoylated. Indeed, when we mutate these cysteines to serines, the palmitylation of CNA beta-1 is abolished. And as expected, the palmitylation of CNA beta-1 is required for its membrane association, because you can see here that mutating each cysteine alone or together abolishes the membrane association of this isozyme, and the mutants are now cytosolic. So we thought that this unique localization of CNA beta-1 must confer the substrate specificity for this isoform, where it can now access to distinct substrates at membranes that the canonical isoforms cannot. To identify the proteins that specifically interact with CNA beta-1, we performed affinity purification coupled to mass spectrometry. And this is a dot plot analysis of a substrate of proteins that were identified to interact with CNA beta-2, beta-1, or CNA beta trunk that did not have the C-terminal tail. Supporting our hypothesis, the proteins that were found to be enriched to bind to the CNA beta-1 isozyme was membrane associated. And very interestingly for us, 4 of these are known to form a functional complex that mediates the recruitment of the cytosolic PI4 kinase to the plasma membrane, which is important for the synthesis of phospholipids critical for calcium signaling. EFR3B is the palmitoylated plasma membrane anchor of this complex, whereas FAM126 and TTT7 work as the shuttle that bind to and recruit PI4 kinase to the EFR3B at the plasma membrane. The PI4 kinase is required for the replenishment of PI4P and the PIP2 pools at the plasma membrane, which are consumed during ligand-induced GPCR signaling. And this replenishment of the PI4P and PIP2 pools are required to sustain this calcium signaling. So we have not only confirmed the enriched interaction of CNA beta-1 with this complex, which was suggested by the mass spectrometry results, but also identified a motif in FAM126 that directly interacts with this CNA beta-1. Calcineurin recognizes its substrates via 2 short linear motifs, and we identified a sequence in FAM126 that fits the consensus of one of them. And using proximity-based labeling, where with the CNA beta-1, which is fused to the promiscuous biotin ligase BRA, we saw that this construct was able to biotinylate each member of the complex. Mutating the calcineurin binding motif in FAM126 resulted in a significant reduction in the biotinylation of not only FAM126, but also all other members of the complex, which suggested to us that FAM126 mediates this interaction between CNA beta-1 and the rest of the complex. We further examined the phosphorylation state of FAM126. And we wanted to determine if FAM126 is actually a novel regulator of the calcineurin, a novel substrate of calcineurin. So very interestingly, the calcineurin binding mutant of FAM126 showed slower migrating bands on an SDS page gel, which we determined to be due to hyperphosphorylation because they collapsed upon treatment with the non-specific lambda phosphatase. We also found that FAM126 phosphorylation takes place at the plasma membrane because this hyperphosphorylated forms of the calcineurin binding mutant of FAM126 is only observed when expressed with other complex members, especially of EFR3B, the membrane anchor of this complex. So in order to study the dephosphorylation of FAM126 by calcineurin, we have to identify the kinase, which is what we're currently working on. But we first wanted to test if calcineurin has actually a role in the activity of this complex. So we hypothesized that during signaling from G protein coupled receptors, the elevated intracellular calcium activates CNA beta 1, which in turn regulates this complex to promote the necessary replenishment of PI4P and PIP2 pools to maintain the necessary sustained signaling. To investigate this hypothesis, we utilized the breath-based assay that was developed by Peter Varnay and Thomas Bala that allows us to detect changes in the PI4P levels at the plasma membrane during GPCR signaling. Using this system in HEX cells that are stably expressing the G protein coupled muscarinic receptor, we monitored the changes in the PI4P levels at the plasma membrane upon induction with the ligand carbachol. And very excitingly, the increase in the PI4P synthesis that we observed in the control cells shown in blue in response to carbachol addition was significantly attenuated in the cells that were pre-treated with the calcineurin inhibitors FK506 and cyclosporin, which is shown here in red. So we are now following up on this to determine the mechanism by which calcineurin is promoting the PI4P production. So to sum up, I have shown you that an isoform of calcineurin has a unique C-terminal tail, and this tail gets palmitoylated. This palmitylation confers distinct localization, access to different substrates, and unique signaling functions to this isozyme. And we also believe that we have identified FAM126 as the first physiological substrates of this isozyme through which calcineurin may be modulating the critical production of the critical signaling phospholipids at the plasma membrane. I did not have time to show you our data, but we have also determined that beta 1 palmitylation is actually dynamic, and it shuffles between palmitoylated and unpalmitoylated states in the cell. The dynamic palmitylation is a critical regulator of many signaling proteins, such as RAS GTPases, whose localization and thus activity depends on its palmitylation state. So we think that the dynamic palmitylation of CNA beta 1 is a novel regulator of its function because its access to substrates at different compartments depends on its palmitylation state. So we are currently investigating the mechanisms by which CNA beta 1 palmitylation dynamics are regulated. And we're also investigating whether the palmitylation of the autoinhibitory tail actually activates the enzyme, especially at the membranes. So with that, I would like to acknowledge and thank everybody who has helped me with this project, especially my advisor, Dr. Martha Sayert, my lab mates, and our collaborators. And thank you for listening to me. I'm happy to take any questions.
Thank you, Idil. That was a very nice talk. There are a couple of questions. One of them is, does this isoform stoichiometrically associated with CNB and its affinity for CAM different than canonical CNA? Yeah, so it has been tested both in vitro and in vivo. Both the purified CNA beta 1 construct and in the cells are able to interact with CNB as the canonical isoform can. There is another question. Is there a cellular phenotype for overexpressed and unpalmitoylated form of calcineurin?
Not that I have seen in my cell lines, human cell lines.
And one final question. If you replace the palmitylation sites with the RAS CAAX box, can you drive even higher P4 synthesis?
That is a very interesting and very exciting question, and I want to test right away, but I don't know yet. But yeah, we have thought of various techniques, maybe with like rapamycin-induced recruitment of the canonical isoforms to see if that would also increase the PI4P production, but we have not done it yet.
Thank you very much.
Yeah,
all right. Thank you for giving such an exciting talk.
Okay.
Our next speaker is Ravi. Ravindra Andral, and he will start sharing his screen shortly. Let us know if it's working. Anyway, in a minute, he will talk to us about the structural framework for the G protein selectivity of muscarinic acetylcholine GPCRs. Okay, take it away, Rory.
Are you able to see the screen?
Yes.
Okay, great.
Put it in slide view, please.
Yes. Okay, first I would like to thank the organizers for this chance to present some of our work that we have been doing in our lab. So today I'll be talking about a structural framework for looking at G protein selectivity in a special subfamily of GPCRs called muscarinic acetylcholine receptors. I'm Ravi Abrol. I'm a faculty in the Department of Chemistry and Biochemistry at California State University, Northridge. And this work was done by a very bright undergraduate that I had in my lab, Luis James Santiago, who is also currently in the audience. So first I would like to, so now you can see Luis, what he looks like. I'd like to acknowledge obviously Luis's contributions and also the other lab members that took part in the discussion, and a few of my collaborators with whom I'm working on either muscarinic GPCRs or some of the other GPCRs that we have projects on in the lab. And the project was funded by a new investigator grant and also more recently with some NIH grants. So to orient the audience, I'll just go through a very quick Biochem 101 of GPCRs in case someone is not familiar with it. Idil talked about some of the downstream signaling that comes after GPCRs are activated. So now you will see some of the upstream consequences that have to occur before calcium or some other signaling pathways are activated. So GPCRs, I introduced them in my biochem class as cell sensory organs because they sense what's outside the cell and transmit that signal somehow inside the cell. And that's mainly done through a conformational change. And these receptors are important in our day-to-day lives that you can actually see and feel because they're also involved in our own senses. So in our sight, in our taste, in our smell, these are all GPCRs. And these are integral membrane proteins, and they derive their name from being able to couple to a class of cytoplasmic proteins called G proteins. And there are 4 subtypes, G alpha I, G alpha S, G alpha Q, and G alpha 1213. And these G proteins are activated in humans at least by about 800 plus GPCRs. And these are important therapeutic targets because about 35 to 40% of the drugs in the clinic target these receptors. So our lab is focused on using structure as a bridge between understanding how a specific sequence of a GPCR translates into its specific physiological function inside a cell. So we're trying to use computational biophysical methods in the lab and structural biochemistry and bioinformatics to understand how GPCR structure leads to their pleiotropic function in the cells. Over the past 25 years or so, there has been amazing progress in the signaling of GPCRs that is captured by a cartoon on the left, which shows that they obviously activate G proteins through the pathways on the left, but they also activate beta arrestins that have their own G protein independent signaling that occurs. And the B panel here shows the time scale at which those signalings manifest themselves when you look at cellular output. So G protein signaling is quicker, faster. That's the first thing that usually gets activated. And then on a long period of time, you also see arrestin-mediated signaling. Now, one of the questions that comes up with many GPCR families is So GPCRs are activated by specific ligands. So here shown with the hormone H at the top. So I'll take the example of a hormone dopamine. So dopamine hormone, and this is the same way that many of the signaling in GPCRs have evolved, is a specific hormone like dopamine can actually cause opposite effects inside the cells, depending on the context. So that's why I call signaling is very context dependent. So to give an example, so dopamine ligand activates 5 dopamine receptors, and 2 of them, D1 and D5, they couple to GS protein. So they actually increase cyclic AMP in the cell. Whereas the other 3 dopamine receptors, like D2 and D4 and D3, they can be GI coupled that decrease cyclic AMP. So the same ligand can actually increase or decrease cyclic AMP inside the cell. And that is enabled through it activating different receptors. So different cells usually may express different dopamine receptor subtypes to enable this. So, but it's interesting from the ligand's point of view how the same ligand can actually activate different receptors and different signaling pathways inside the cell. So these receptors obviously have evolved together as a family because they are all activated by dopamine. However, even though their structures are similar, they still activate different G proteins. So the research that I'm going to tell you about is trying to understand the structural basis for this signaling in the context of the other ligand, which is called the acetylcholine and its receptors. So over the past, I would say, 13, 14 years, there has been amazing progress in the structural biology of GPCRs that led to the structures originally of isolated receptors, and then later on receptors bound to either G proteins. Here on the left-hand side is a full G protein bound to beta-2-adrenergic receptor that came out of Kobilka lab. And then the structure on the right that was rhodopsin bound to beta-arrestin. So we now know these receptors, what they look like when they're bound to their signaling partners. And over the past few years, thanks to great strides in cryo-EM methods, now we're also getting structures for receptors bound to different G proteins. So here's an example from Kobilka and Skiniotis lab from Stanford, where they last year obtained structures of the 2 muscarinic receptors and bound to their cognate G proteins. So the M1 was bound to G alpha 11, that is from the GQ family, and M2 was bound to the GO protein that is from the GIO family. So these structures or these structural snapshots, they're providing us great starting points in order to allow us to understand now the structural basis for why these 2 different receptors, for example, activate their cognate G proteins. For example, why M1 will not activate GIO and why M2 will not activate, let's say, G11. So now focusing on the receptors of our interest, so we will be looking at muscarinic acetylcholine receptor family that is activated by this ligand acetylcholine. It's involved in many, many different physiological processes in our bodies. So this ligand activates 5 receptor subtypes that are GPCRs, and 3 of them couple to G alpha Q, and their downstream signaling is shown here in the box. So they all increase activation of PLC beta, increase DAG, IP3, increase calcium levels that Idil was talking about, and also they increase the activity of ERK1, 2, and JNK. On the other hand, the other 2 receptors that acetylcholine activates, M2 and M4, they actually couple to GI proteins, and they, for example, lead to lowering of cyclic AMP, but they still increase the activity of ERK1, 2, and JNK. So we see that the same ligand can activate different pathways and same pathways. So we are trying to understand here how this occurs at the atomic level, at the receptor G protein interaction level. So in order to do that, we use the structures from Kobilka and Skiniotis lab from Stanford, the cryo-EM structures of M1 and M2 as starting points to build homology models of these 4 complexes. So, so we build the complex of the cognate complexes, which is M1 with GQ and M2 with GI. But we also built the non-cognate complexes, which are much harder to study experimentally because these are membrane proteins. So computationally, we can easily build those. So we also built the complexes of M1 with GI that are not seen in cells, and also M2 with GQ that are not seen in the cells. And we will follow these 4 complexes through these computational biophysical methods to see how stable they are and what can we learn from these structures in terms of the G protein selectivity of the cognate complexes. So in order to do that, we relax these proteins in their membrane environment shown on the bottom. And we did that for about 2 microseconds for each of these systems. And then after that, we did a free energy analysis of the interaction energy between the receptor and the G protein. And we did that using this molecular mechanics based method called MMPBSA. So I won't have time to cover this method, but you're welcome to ask a question later on and I can talk about it. But in a nutshell, it captures the solvent as an implicit environment, and in that it allows us to calculate the free energy of interaction between any 2 proteins. In this case, we are using the receptor and the G protein, and we do this for all 4 complexes. So first we look at the relaxed structures of the receptor. So in orange and blue are the M1 and M2 structures shown after the MD simulations, and they are compared with the starting structures of that came from the crystal or the cryo-EM templates. And they were off by about roughly 2 angstrom in RMSD. So they're very similar to the starting structure. We looked at the ligand binding site, and the ligand binding site was quite stable. So the binding site residues around this I-peroxo ligand that was bound to these complexes remained stable. The ligand moves a little bit based on the complex, but overall the binding site is pretty well maintained during these simulations. So next we looked at how the G protein oriented itself during the simulation, and that you see here by looking at time t0 shown on each of the panels, and then what happened at the end of the simulation. So in all cases, the G protein moves a little bit from the starting point, because this is the whole idea of doing these simulations and relaxing these complexes in the native environment, because the cryo-EM provides as close to native environment as possible, but it still can have sort of effects coming from low temperature and packing of these highly concentrated protein solutions. So the simulations shown here, they allow these protein complexes to relax and find their natural orientations. And also we are building these complexes from the cryo-EM structures. So some of these helices, like shown here for the G protein interacting with the receptor, move a little bit, but they are overall very stable. So these are the final complexes. So on the left-hand side, we are looking at M1 receptor bound to either GQ or GI. And on the right-hand side, we're looking at M2 bound to GQ or GI. And the big difference you see that on the left-hand side is that when the M1 is bound to GQ and GI, the GQ and GI are in 2 different orientations. And you see roughly an angle of about 30 degrees between how the G protein is oriented with respect to the receptor. And that comes from individual local interactions that are keeping the 2 proteins together. On the other hand, with the M2 receptor, the GQ and GI are more close to each other in terms of how they interact with the receptor. But overall, all 4 complexes were stable during the simulation. So then we looked at the free energy of binding of the 2 receptors to the 2 G proteins. And that result is shown in this average row. So you're looking at these energies in kilocalorie per mole. And what we find from this analysis is that the cognate receptor G protein complexes shown in the green boxes do have the most stability, which means that they have the strongest energy of interaction between the receptor and the G protein. That's shown in comparison, for example, of M1GQ with M1GI. M1GQ has a stronger affinity compared to M1GI, for example. And this result is actually consistent with previous studies on the M3 receptor, which we did not study here, with the M3 receptor, where they actually showed that the thermodynamics of the interaction is the predominant factor in determining the selectivity. And kinetics is not a main factor, because when we started this project, we were worried that it could be both kinetics and thermodynamics that might be playing a role in terms of selectivity. But at least in the case of muscarinic receptors, we have shown now that the thermodynamics dominates the interaction between the receptors and the G proteins. And if there are any kinetic effects in this association, those are weaker and they don't affect the selectivity. However, that might not be true in other G proteins and other GPCRs. This study is also consistent with a computational study that looked at the receptor and just the G alpha 5 helix interactions, and there also they found that the thermodynamics played an important role in the interactions.
Hi, Ravi. Sorry to interrupt, but we are approaching our time limit.
Okay.
So if you just want to have one closing sentence, and then we'll have some questions.
All right. Yeah. So this is essentially my close to the summary slide. So we looked at the interactions between the receptor and the G protein to figure out what were the selectivity determinants, and we do see that in at least M1 we have more interactions and they're also more stable, whereas in the case of M2 with the GI, we have less number of interactions, but overall they're more stable, or they're stronger. So that helps to understand the selectivity. We do find in the case of both the cognate complexes, M1GQ and M2GI, this conserved TM5, TM6 contact that is present in the receptor that is maintaining that cognate receptor G protein complex. And we'll be testing this coming year by doing mutational studies to figure that out. But overall, these studies show or support what's called a barcode hypothesis for G protein selectivity, where it's not individual residues that appear to be responsible for G protein selectivity, but it's a combination of those residues, and those residues are spread across the entire structure of the receptor, and because of receptor being very flexible and a lot of allosteric effects going on in this interaction. So this barcode hypothesis makes sense in terms of understanding G protein selectivity in these systems. So, and if you want to look at this data more closely, you can look up this paper, and I'll be happy to answer any questions.
Fantastic. Thank you so much for such a comprehensive talk. There is one question. Would including the beta gamma dimer of the G protein in the simulation affect the result? So
we are following up on that. So in this case, we looked at the full G alpha protein bound to the receptors. In some of the previous simulation studies, people have just used the G alpha 5 helix in the alpha protein. So, but our future simulations will be taking the beta gamma into account. And I believe that at least in the case of muscarinic receptors, it should sort of enhance the interaction energy and corroborate what we have found so far.
Interesting. And another question, have you explored the number of contacts in each of cognate and non-cognate cases?
Yeah, so that's actually captured in this image where the size of the circles here represents, for example, how many contacts TM6 on the receptor side makes with the G protein. So in terms of number of contacts, for the M1 receptors, the number of contacts is bigger for GQ versus GI, and that explains the cognate selectivity. Whereas in the case of M2, the number of contacts does not explain it, because you can see here that TM6 and TM3, they make more contacts overall with for M2 with GQ, but that's not the cognate receptor. But in this case, the number of contacts is not enough. The strength of those contacts is equally important. So in terms of strength of contacts, the M2 GI wins, and that is the cognate complex.
And finally, there are several questions that asked about the membrane.
Yes.
What lipid bilayer did you use? Have you considered varying lipids, specifically which part of the membrane?
Yes.
Yeah.
Yeah, so we have in this simulation system, we used, I believe, POPC, lipid bilayer. We have not varied the lipid composition. But if there are studies that show that the lipid composition affect M1 and M2 receptor coupling with the G protein, would be definitely love to do those simulations.
Thank you so much. That was a very good talk. And
Thank you.
thank you to all my fellow panelists here, Idil and Ravi. And thank you all for joining us around the world under these circumstances. Thank you to ASBMB for organizing this session. I wish you good health to you and your families, and we will conclude the session now.